Greetings and welcome to Repligen's 2021 Annual Technical Seminar. If you've attended a technical seminar in the past with us, welcome back. If this is your first event with us, we hope you enjoy the next few hours and we hope to see you again in the near future. My name is Wesley Straub and as the Marketing Director here at Repligen, and it is really my We love questions. To access our Q&A toolbox, just use the 2D barcode down below and it opens up a small window. In that window, you can type in any question at any time, and we will field those questions at the end of the series. Okay, because we have 5 talks today, we will limit the Q&A period to 5 questions. In order to manage that a little bit better, we highly recommend that you use the upvote function in the Q&A. Little upvote. So if you like a question, give it an upvote, and we'll do our best to connect that into the conversation. Okay. Okay. Done with housekeeping. On to this year's topic. Each year, bioprocessing is a little different, and our technical seminar series addresses a specific topic. This year, we feel is a little different than most. Typically, we focus on a new application or maybe a new technology. But due to the confluence of the first pandemic in nearly 100 years and the rapid approval for multiple gene and cell therapies, the landscape for capacity and supply chain have dramatically changed. Not just the amount of capacity that's required in bioprocessing, but the kind of technologies that move through that capacity. And therefore, this year, we focus simply on the capacity challenges and how Repligen is helping to overcome some of those challenges. Additionally, I think on top of that, and led by a courageous few, the need for equity in our cultures has reached a certain decibel. So it's not only capacity for the new vaccines or gene and cell therapy, but it's capacity and cost reduction and flexibility across the biopharmaceutical portfolio that needs to be addressed. So altogether, these multiple driving forces have created multiple needs, which are articulated with both inspiration and aspiration by our plenary speaker, Chris Love, who's the professor of chemical engineering at MIT's Koch Institute. In discussion with Chris and all of our presenters during the preparation phase, the group strongly aligned towards Chris's emphatic use of the word "and." So while the title of today's seminar series is Overcome Bioprocessing Capacity Challenge, it might also be titled Overcome Bioprocessing Capacity Challenges and, because it's not just one thing anymore. Solutions that really move our needle need to address more than one facet of the current manufacturing challenge. So with that, here is Chris Love, our plenary speaker, to kick off the 2021 Repligen Technical Seminar.
This last season has emphasized to us the challenges in manufacturing in a timely manner for biopharmaceuticals and vaccines. Let's take a few minutes to talk about how we might see transformations in the type of manufacturing that we enable and the nature of it as we look to accelerate both for pandemic readiness and for normal business in advancing new medicines for patients. In particular, I'd like to take the next 35 minutes to think a little bit about what are some of the factors that we've seen from this past pandemic season that have helped identify challenges in manufacturing systems, what are some of the considerations that we might want to take in enabling new strategies for that manufacturing? And what are some of the technological innovations that are enabling next generation manufacturing as we look ahead? The pandemic has highlighted for us a number of different challenges. Just sheer raw capacity for manufacturing biopharmaceuticals and vaccines, and the numbers needed to treat large numbers of patients across the planet, has really highlighted how these facilities that take time to build, qualify, and operate can be limiting in these types of scenarios. We've also seen supply chain logistics start to impact our ability to deliver timely medicines in not just COVID-related interventions, but other normal medicines as well. Everything from plastics to steel to chemicals are things that are affected by the current supply chain challenges that we see. And then finally, there are a number of parts of the world that are only now beginning, if at all, having access to many of the interventions that those of us in high-resource countries enjoy already. This highlights the challenges in how do we make drugs that are available for everyone. If we consider the totality of the manufacturing capacity today, this analysis was done just prior to COVID, about a year ago, and identified about 6.5 million liters of capacity. This capacity is split among mammalian hosts and microbial hosts and produces a large number of drugs in various stages of clinical development and commercial use. A lot of this manufacturing is centralized in a small number of manufacturing facilities around the world that take advantage of these capacities to make these products for innovator companies as well as CDMOs. If we kind of run some numbers to estimate how much drug capacity these manufacturing centers could produce, if all things were equal and all processes the same, we could estimate on the order of 10 to 100 metric tons produced annually out of this kind of capacity. This serves, maybe 10 to 200 million patients globally for the 200 or 300 indications represented by the drugs that are listed here. These numbers highlight in a sobering way the capacity constraint that we see today as we're trying to bring interventions to the world for vaccines and therapeutic interventions that would address COVID-19 specifically. The manufacturing capacities that we have require a variety of different fits of the processes that they enable in order to operate. So we're even further constrained in that these facilities are not all interchangeable in the ability to make a wide range of different products. All of this constrains our overall capacity for bringing new medicines online. And as we've seen in this last season with the response to COVID, remarkably, there's a number of different manufacturing technologies that are needed to advance not just COVID interventions, but as we look ahead, a whole range of new modalities that are present and now emerging very rapidly as a result of this past season. Examples, of course, include mRNA, but also live viruses that are engineered to produce specific responses or manipulate genes in a targeted manner. And while we've not seen cell therapies as part of the direct response to COVID, this additional modality creates another type of biologic for which manufacturing solutions are going to be needed. So our challenge is not just capacity, but the diversity of the use of that capacity to be able to produce the types of interventions that might be important for COVID, future pandemics, but all of the other indications that we also are developing therapies for. All of this kind of highlights aspects of what may be really critical as we think about the next generation of biomanufacturing solutions that go beyond the current capabilities. A lot of times when we talk about the types of manufacturing qualities that we're interested in, speed, costs, flexibility, or simplicity, we're often asked the or question. You can have 2 or 3. But as we look at what's needed to respond in situations like this last season, as well as the ability to rapidly bring new therapeutics to market as we have better understanding of the biology, this becomes an and. We need speed and low cost and flexibility and simplicity to minimize those supply chain requirements. So we need to think about the innovations in technology that will enable this and in the next generation of manufacturing. I want to take a moment to look back a little bit, to think about another industry that has gone through a similar transformation, one that perhaps highlights what might be ahead of us in the manufacturing of biologic medicines. The printing press was a remarkable invention that allowed for the dissemination of information through printed pages, but it was really the advancement of continuous manufacturing strategies, like the one that's shown here, in the early 20th century that enabled reel-to-reel production for newspapers, magazines, and a large number of publications across the planet. These facilities, while expensive, were efficient in being able to bring forward that information at a scale that hadn't been possible previously. In fact, if we kind of look at what the impact of continuous manufacturing was on the number of pages printed per day, we could see that through the traditional printing press technologies, on the average of 10% to one full page per year per person across maybe 1,000 printers globally when the printing press was first enabled. But with the advent of offset rotary printing, it allowed for now more than 300 pages per person per year. This continuous manufacturing really facilitated the transformation and the ability to now manufacture at a much different scale than what had been previously possible. I want to take a moment here. We know that in our industry, the continuous manufacturing is becoming more centralized in our innovations that are coming forward and touching a number of different products. We see this most particularly in the context of monoclonal antibodies, but increasingly in cell-based therapies and other areas where the benefits of continuous production can have an impact on the ability to manufacture in an efficient manner. But I want to think about the evolution of this industry a step further. This is a Google search on inkjet printers. Many of you probably have one at home. I have one sitting here next to me. This technology allows anyone with a printer to produce a printed page, and to do so in color on a variety of different formats and with high quality, such that these can be publication grade quality prints, and all for about $100. The Xerox printer was really the first advent of this, and it came along about the same time that continuous manufacturing was being really truly transformative in the commercial printing sector. Early days of this type of technology was not able to produce at nearly the same scale as these other types of more advanced, capitally intensive technologies. But with the evolution of that solution for small modular manufacturing of printed pages, suddenly we're at a state today where more than 100 million printers ship annually to produce a total amount of printed pages that is comparable to the continuous manufacturing solutions that we enjoy in conventional manufacturing plants. That is, that the capacity gap is closed due to the ability to now distribute that manufacturing with high quality in a variety of different locations. There's some serious implications here for the impact of this kind of technological advance for manufacturing. The demand for expanded access to product, in this case, printed pages, has promoted the commoditization of the manufacturing technologies themselves. Even a step further, we can think about 3D printing as another level of customization of unique products that are made through a manufacturing technology that is becoming commoditized. It's the intensification and integration of the parts that have been essential drivers for this scaling that have enabled the cost targets to be reached. A printed page on an inkjet printer is approximately the same price as that made in large facilities. And it has traded off the capital expenses for operating expenses. But what this more broadly enables is a diversity in solutions. Anyone is now able to be their own publisher. Now, while this might be a bit far-fetched to think about for manufacturing of biologic drugs with the appropriate quality and requirements for oversight that we have in this particular industry, there's a lesson here that I think we can appreciate, that technology can facilitate and advance that transformation that we should look at as we think now about the and. How do we facilitate manufacturing capacity building that features low cost, speed, flexibility, and simplicity? Let's take one more example and look at this a little more close to home. Centralized manufacturing solutions for vaccines, which are incredibly important in the current pandemic, historically have failed in the highly centralized models. Every year, the WHO and others set targets for global coverage of vaccines of many childhood diseases. And every year, those targets are missed because of failed batches, because of long distribution routes that lead to uncertain product in different locations. And all of this has led to sort of a stagnation in the overall coverage for a number of different vaccines. And that's to produce the vaccines that we already had, not the new ones for COVID. The global capacity for manufacturing of vaccines has been about 5 billion doses per year for all vaccines. For COVID, in this last year, we've been aiming to produce 19 billion doses. There's a pressure to be able to manufacture at that scale, and centralized facilities that require bespoke processes and infrastructure have been limited in their ability to bring that forward historically in other vaccine contexts. Returning to our model of printed pages, distributed manufacturing networks, where there is more regional capabilities with more standardized facilities, might allow to tailor the manufacturing capacity to meet the demand required, and could ultimately democratize the vaccine supply itself. Different regions could manufacture vaccines according to their needs and for the diseases that are endemic in their regions. And in global pandemics, this capacity could be utilized to facilitate global solutions. So let's dig into this particular idea a bit more. If we think about what's required, and I'm going to use the example here of recombinant subunits or recombinant, well-defined, well-characterized biopharmaceuticals as one example of the modalities that one might make. We want to be able to produce a wide range of different molecules in some kind of manufacturing solution, and we want to be able to do so quickly with speed and efficiency. But all of those things that we manufacture should retain all the features that we expect of a high quality product. The identity, purity, safety, potency, all the features that are the quality attributes of those products. At MIT, over these last 10 years, we've had an opportunity to think deeply about this question as to how the technologies that have now been in the forefronts of advanced processing for continuous ideas start to manifest themselves in a modular, fully connected, and fully automated solution to produce something like drug substance. We produced a benchtop unit, like the one that's shown here, in kind of a prototype version that comprises 3 different parts. A production piece that relies on microbial perfusion, so perfusion of the material to allow for a small volume of that production, and microbial systems to allow for speed and flexibility in the production of different biomolecules. That material is then processed in a purification stream that takes advantage of continuous batch processing, but does so in a unique way, leveraging integrated straight through bioprocessing. We allow for a single point adjustment, and then the modification of the eluents of each stage leads into the next one to allow for kind of continuous and operations of this particular module. It starts to blur the lines between the conventional stages of chromatography such that this unit performs the entire totality of the objectives desired: the reduction of host cell proteins and DNA, and the purification of variants that may be present in the biomolecules that are being made. And then finally, a formulation step that allows for the exchange of the buffer for storage or other use, or direct use in non-clinical studies, as some of the examples that we've demonstrated here. Using this exact system, we've now produced more than 12 different molecules of different types: hormones, cytokines, vaccine components, not just a single class or a single type of molecule, but a wide range of molecules in this same platform. And taking advantage of all that speed and continuous operation means that our volumetric productivities can rival those of commercial monoclonal antibodies today, but now for a wide range of non-monoclonal products. Taking the same idea a bit further, this is a printer scale system. It's a benchtop system. And while this is great for prototyping ideas, a simple scale up to roughly a few liters of reactive fluid in a perfusion model at the scale of sort of an American deli refrigerator allows for the production of roughly 10 kilograms per year per system. This is fairly remarkable when you consider the number of doses that this translates into. One system annually could produce enough vaccine for like the state of California, or for a whole country. A few of these could produce for an entire region. This is pretty dramatic when you consider that this is effectively a piece of equipment. We've taken a process that we've built on the printer scale system and used it on the refrigerator itself, the refrigerator scale version, to produce GCSF as one example. GCSF is a first generation biopharmaceutical used in the treatment of cancer. We've taken the same process and directly transferred it into the larger scale system and produced material of the same quality with the same features and speed that we had at the smaller scale. This allows for a direct transfer of operations from one to the other. The same design and architecture allows for simplified tech transfer between these units. And using the same kinds of bioprocess, we can make a wide range of different molecules. These are just 4 examples of processes that are run using the same systems for 2 or 3 stage chromatography operations that allow for the production of a wide range of different molecules, all of which produced microbially today would require bespoke processes and often customized facilities to house and enable each of those. So now, using a similar common platform, we can make a wide range of molecules with phase-appropriate development qualities in the demonstrations that we have shown to date. But what about cost? Working with Susie Farid's lab at University College London as part of a grand challenge with the Gates Foundation, we've asked the question, what does it look like to incorporate these kinds of innovations into cost models? What might be possible if we broke down the large centralized manufacturing to focus on smaller, more modular facilities? The levers that we find are the same ones that we would appreciate in other contexts. Intensification of the process through volumetric productivity and reducing downstream operations helps reduce the cost. Automating these solutions to be fully hands-free, as the systems that we've shown here are, allows you to further reduce the OpEx costs that are associated with the operation of a facility. And the simplicity of these modular facilities allows for much more straightforward layouts in the kinds of ballroom-like styles that we've seen emerge in biopharmaceuticals today. All of these features, when combined with a multi-product by design facility, can let us hit a cost of goods for a drug substance vaccine component that exceeds the cost target that we have been given as a challenge, about 9 cents per dose for only 40 million doses per year. This suggests that it's possible to enable micro facilities for vaccine production, but also other biopharmaceuticals that can be cost competitive with large-scale centralized manufacturing, as we incorporate in more innovations in process intensification and automation. But another interesting thing to consider is where else do we have levers to facilitate reducing timelines and costs in manufacturing, particularly of protein biologics, but of any biopharmaceutical of interest. I've listed here a number of different factors that impact the cost and the time to bring something forward, including the productivity of the host, the yields from our process, the quality metrics required, and the CapEx and OpEx required to build the facility and operate it around the process that we've built. Interestingly, the common denominator in all of these different factors is the choice of the biological expression system. So as we look at the implications of what's happening with integration, automation, and intensification of manufacturing processes in this current era of biology with gene editing and genome sequencing, it's now possible to include the biological expression system in that equation to maximize our manufacturing throughput on reduced timelines and cost. Shown here is just one example of how the expression system can shape the complexity and operational timelines in a facility. I've picked two examples of an E. coli-based process and a CHO-based process, and canonical monoclonal antibody process. And then here in this example, Alan Coleman, along with Amgen, looked at what would it look like to bring an alternative host like Pichia pastoris into an equation like this, and where could you start to see savings in your operational state? I won't have time here to go into all of the details, but you can see that we're reducing the number of operations required as a factor of both the speed of the host and the biological capabilities of it. For example, its inability to harbor adventitious agents. All of these things lead to increases in the volumetric productivity and the reduction of buffers. So we're increasing the productivity in the volume of the facility that we're using. There's fewer steps, which reduces labor and raw materials. And all of these steps that are now simplified are amenable to the kind of integration and automation that I've already shown you. So in this way, the biological traits of the host itself can unlock these simple intensified processes that go a step beyond where we are today in our manufacturing approaches and platforms. So I want to spend the last few minutes here to focus on this element of what does it look like when we start to engineer the host itself? How can we improve the host to facilitate the kinds of advances in manufacturing that we've just described? About 2 years ago, 3 years ago now, we had an opportunity to start a consortium with the industry to think about this in an intentional manner. How could we begin to engineer hosts for this next generation of manufacturing that enable flexibility and low cost and speed and ultimately simplicity? How do we engineer that host to facilitate this, and what are the best hosts for doing so? To begin with, we started with a yeast organism, Pichia pastoris, because it has a well-established track record in manufacturing biopharmaceuticals and vaccines today. But these ideas are not limited to this. They could be expanded to any number of hosts with this principle and approach. I've listed here a few different factors that we might want to be able to engineer to produce something like a monoclonal antibody in Pichia pastoris. We want the parts to be able to mix and match the vectors and the tools for editing the genome, but also it needs to have the features that make it amenable to high intensified processes. Fast growth needs to not use things like methanol that can challenge the designs of facilities, and it needs to have productivities that will compete with the best of what's available today for monoclonal antibodies or other proteins. It also has to produce the molecule of the right qualities. What we're thinking about is how do we do this from a first principle, biologic data-driven approach to engineer the host fit for purpose to enable these types of intensified processes. I won't have time today to talk about all of the details of things, but just a couple of examples of how we're thinking about this to integrate the engineering of the organism itself as part of this intensification of the manufacturing processes. Host-informed genome editing tools like CRISPR-Cas9 can facilitate very straightforward editing of genes in these organisms. This makes it possible to do in a few days the types of things that might have previously required weeks or months to enable. Things like knocking out genes that are associated with the glycosylation pathways in the yeast organism. We can now do this with extreme efficiency to cycle through different designs and iterations of concepts based on the biology that's informing them. One example of how we might use this type of tool is to remove the methanol dependency that's often found for production with Pichia pastoris. We've used RNA sequencing to help inform how the cells respond in the presence of different process conditions, like a carbon source for methanol or an alternative one like sorbitol. What are the pathways in the biology that are being impacted, and what genes specifically are being activated in the presence of methanol that we could supplement and replace through targeted gene knock-in into the host to avoid using methanol but get the same benefit in sorbitol. Shown here are some examples of activating transcription factors that can be knocked in, and you can see the impact here on production. In the 5% methanol case, this is the typical time scale of a fed batch process that you might conventionally run with a microbe. It's about 120 hours, and you can run that system up and it will ultimately crash as that system can no longer maintain the best biological state in the fed batch system. But removing methanol and the oxidative stress associated with it in the context of running perfusion-based processes, like the ones that we've shown here, we can get sustained production of, in this case, vaccine antigen for rotavirus over 220 hours. These are extensively long times. And in fact, some of our perfusion processes in the past, we've operated for several weeks. This changes the game. We have an ability to use microbial systems that are fast in starting and engineering in a way that we can use them with the best of intensified continuous production today. Another example of further process intensification through host engineering is taking that same host and engineering it now for something that might be, for example, a trivalent vaccine candidate for rotavirus. That is, it needs to have the vaccine components, needs to require 3 different proteins that are related from different serotypes of the virus. We can think about this like COVID-19 today with different kinds of variants of concern. We might think about in the future multivalent vaccines in that context as well. But here, for rotavirus, we have 3 antigens. Historically, you would make each of these 3 different molecules in 3 separate processes, or 3 times the cost and time required for that manufacturing. Here, we've engineered one strain with promoters that allow for the production of all 3 of these antigens in a way where the biology and perfusion can be maintained to keep the same relative balance of these 3 antigens. Importantly, though, with this straight-through processing that we've developed on the downstream side, we found a process that could purify and retain the balance of those 3 antigens all at the same relative ratios. In this way, you could imagine a single process for producing this trivalent rotavirus vaccine through controlled biology and understood design in the process purification to enable that particular process. This is a proof of concept, and much more would have to be done to advance this, but you can see how host engineering can facilitate this type of rapid production. And in fact, we had an opportunity in this last year to apply a lot of the ideas that we have talked about here to the advancement of candidates for low and middle income countries for SARS-CoV-2 vaccine candidates. And in about a month, we went from a DNA sequence to strains that were ready for cell banking as master cell banks, as well as a draft process that could be enabled and run on the type of system that I've described to you here. Iterating on this allowed us to further refine those designs to make advanced candidates that are similar to some of the variants of concern that are in circulation now. Some of these candidates we've been able to transfer to manufacturing groups who have taken these and scaled them up for clinical stage development of vaccine candidates that are in the clinic in various parts of the world. While this has been an exciting part of the response to the challenge of this last year, It also highlights some of the advances that are possible with this kind of targeted host engineering that is very particular to the biology of it and driven by the principles that we learn in how cells see molecules and how they ultimately produce them. To summarize, we're at an interesting point in time for manufacturing technologies and something that we're going to touch on throughout this session. Modular facilities in the form of ballrooms are getting smaller and more modular by the ways they're implemented. Single-use technologies, plastics, and these types of technologies for all of the different components in manufacturing allow for simplified supply chain logistics in the turnover of these types of modular components. Continuous operations are now becoming enabled because of some of these types of advances. And as a forward-looking and important aspect of this, the ability to read and write biology like software is now allowing us to engineer hosts fit for purpose to be able to enable a wide range of molecules beyond simply monoclonal antibodies as platform-like solutions. So just to share a couple of final thoughts on distributed manufacturing and this future state for production, there are benefits beyond simply pandemic readiness. The ability to take ideas fast from the research lab into the clinic is a key enabler of having access to this kind of manufacturing. At scale, for scale process development also facilitates technology transfers and ultimately could prepare for breakthrough readiness of smaller indication areas where small volumes of commercial production are sufficient. There's a predictable cost of expanding and shrinking markets in your manufacturing capacity through these modular designs. There's a trade-off between traditional CapEx into OpEx of these equipment as you start to facilitate these types of scale-out solutions. Agile supply chains then become a possibility in this model. No longer are we pushing drugs through supply chains to have stocked staged material at each stage, but rather production on demand as required. And then ultimately, this kind of access to manufacturing capacity can improve access and innovation of differentiated solutions that are regionally focused and that have different populations' best interests at heart. I'll thank the folks that are involved in the work. I've talked about several of them as we've gone along, and funding from DARPA, the Gates Foundation, and Allpost, who have helped us think about these different ideas and their intersection for future manufacturing solutions. And I'll leave you with just this one thought. Can these kinds of innovations that we're talking about here today begin to really expand access and equity to manufacturing of medicines that are important for non-communicable diseases? Cancer and all of these other non-communicable forms of disease are common now in all parts of the world. Yet access to many of these medicines remains poor. How can we use innovation and technologies to facilitate regional discovery and regional production that facilitates access to these kinds of medicines and builds a better discovery engine across the globe?
Okay. Welcome back, Chris. Great talk. Really, really grateful for setting the stage with that emphatic use of "and," right? Today's topic is really capacity and several other things, flexibility, cost reduction, so on. Chris, we have 3 questions in our Q&A, and you are more than welcome from the audience to add some questions as we discuss. We have 2 from Finn and 1 from Sai. My apologies if I don't pronounce that correctly. I'm going to take them in chronological order. First one, Finn asks, is there a metric or set of metrics that captures overall process complexity that will allow quantitative comparison to other technologies? Parentheses, printing. So I think
that's an awesome question.
Yeah.
Yeah, no, Wes, I mean, that's a very insightful question. One we actually should think more about together collectively as to how can we better define what do we mean by productivity. At the end of the day, the choice of a host and the materials that we use, all of these things are really just part of a catalytic process that takes those raw materials and converts them into high value products, in this case, biopharmaceuticals or vaccine components. one metric I think we talk a lot about is volumetric productivity, but I don't think we spend enough time talking about volumetric productivity as defined by grams per liter per day per meter cubed of the facilities that we're utilizing. That in part captures some of this complexity that we're talking about here. Less buffer preparation space, more connectivity between operations, more harmonization in the different stages of upstream, downstream, and release, fill finish. These are all ways I think we could start to capture that. But I think it's actually a really good question is, what's the right set of parameters that we want to capture in a score of that type? Is it, the collection of the volumetric productivity plus costs, or that plus a number of operations that have been reduced? I think these are things that we should probably talk more about as a community.
Yeah, because you could add so many more denominators to that. You could add cost to that. You could add FTE. You could add the time it takes to build the facility to get your distributed model, right? You could add into that how much did it cost to get to that facility, and what's the flexibility in putting a plant like that someplace in the globe. I mean, there's so many denominators that you could put to that volume under.
And I wonder too, Wes, maybe it gives us a way to think about comparing where is the biopharmaceutical industry today in comparison to other industries, and what are ideal industries that we might want to see shifting towards, and what are the differences in those metrics between the two. I gave the example of printing presses because I think it's a nice one that kind of highlights the potential to expand capacity in multiple different dimensions. But how do we kind of sit, where are we now and where are we going, I think is a key question.
Yeah, because I like the question too. Because when I read that, it made me think. I went to the car industry, right? I'm like, their process is a little more complicated. It's a regulated environment. Their electronics to update a chip is kind of similar. If you update a process controller on our side, right? So I'm curious, like if they have metrics, their processes are far more advanced than bioprocess, mature, right? What do they look like compared to?
Absolutely. And I only had time to pick one example, but the car industry is another one that I often look at in these conversations, because it's another one where in the modernization of manufacturing there, it has become regional. Cars aren't made globally, they're made regionally to fit to local requirements for those products. And as you say, that's also a regulated industry. So it's another one that I think we could learn a lot from.
All right. Size question got 3 upvotes, so we're going to skip and go to that. Where does the industry stand in the adoption of process intensification, such as the use of membrane bioreactors? So I'm thinking specific about the kind of the modified wave bag bioreactors that have a membrane built into the bag for basically cell retention or perfusion, like in the reactor. Yeah. Any thoughts on that?
Yeah, I think we're seeing a lot more openness to the transition of these technologies into the use of preparing clinical lots as a way to evaluate these innovations in that setting, but also into commercial production. And I think you can look at examples like the integration of the ATF and some of the continuous operations that allows now, moving more towards commercial implementation. Now, groups like Boehringer Ingelheim and Pfizer have developed a strategy for doing so. And you see Sanofi committing substantial resources into the development of a modern facility that takes advantages of many of the continuous bioprocessing strategies that they've pioneered over the years. So I think, we're at a point now where the technologies are changing so quickly, and we were talking a little bit about this before, they're changing so quickly now. How do we facilitate their adoption through this strategy? And I think the model that's developing is a good one, where those technologies get integrated into the pilot labs and the clinical production as a way to kind of vet them going towards commercial and kind of locking down in those ways. And so I think, the interchange of different technologies in that regard will come from just simply the demonstration of their utility in each of those different stages. But there's so many to look at now that I think are quite exciting. And, again, a reason why we should think holistically about the selection of all of the components together to really enable the kind of process intensification I think is feasible now.
Yeah. I mean, I think in some ways so in your talk, right, I mean, you went through all of it. You went from all the genome host side of it, like host selection, edit the genome of the host, and then you mentioned like the advancers like Boehringer Ingelheim, known people who have known to either build completely single-use facilities or like continuous downstream facilities, right? The challenge I have in the industry right now is the other spaces, even not just in the menu, but like for example, the clinical people have gone to cloud infrastructure a long time ago, right? Whereas like getting quality data across multiple sites in the in US and North America, we're still not there. Or the clinic basically took on in the 90s diagnostics as a surrogate to endpoint data for approval of a drug, or even the and these are things that basically, or qPCR for an HIV diagnostic. qPCR in the world of bioprocess, I worked on the mycoplasma technology for a long time, right? That took us 8 years to get into lot release of a product. And so something in our, I think the vastness of possibilities there. I just curious how we can get there faster, because there's a lot out there that I think from other industry can bring in. I just want to do it faster than we've done it over the past 2 decades, maybe.
Yeah, I mean, I think the timeline of kind of 5 to 10 years of adoption of these things makes sense given all of the challenges and components in that adoption process of bringing them along, and to evaluate them critically in the context of the role that they play in the totality of the solutions here. I think though that the technologies are changing so much faster than that process historically has been, and so I wonder if this is a place where a little more cooperative infrastructure among the different partners in this space could help facilitate the adoption of these things. More targeted attempts at bringing forward next generation platforms to facilitate these things. I think speed continues to be something that we're going to think a lot about, not only from the business perspective, but also in the kind of pandemic readiness that we're seeing play out here in kind of live time. I think we have to find ways actually to work together a little bit more cooperatively to bring forward some of these solutions, and perhaps that would be a way to help accelerate it. I look at consortia like the multi-attribute method consortia, or the alt-host consortium that we've formed at MIT. These are examples of ways I think that the industry can get together in a pre-competitive way and start to talk a bit about what's it going to take to realize those transformations.
Yeah, exactly. Last question, second question from Fim, and then I'm going to thank you for your time and transition to the next part of the seminar. Is, does the prototype setup, I think the benchtop setup that I saw with a couple different technologies, does that include analytics?
So it has a number of different process analytical technologies in place for traditional process data. And I think one of the things that, again, comes back to this holistic consideration, our experience in using this host is very predictable. And when it operates within its process specs, it creates products that meet the quality attributes that we anticipate that it's going to have. And there is an understanding within the regulatory environment in Europe, for example, to allow for this type of parametric release based on the ideas that are there. I think the difference is when you have these benchtop systems, instead of running that facility maybe 3 times a year, because that's all you need for that large scale element, we can run it 50, 100 times. It depends on how many units you have, but your ability to accumulate that data and demonstrate the relationships, I think, is quite different. So I think as we look at what process technologies would be critical for release, these are ones where we can be very thoughtful about it now, considering the totality of the data that's going to be available.
Yeah, I can definitely see that. Like running a bioreactor 3 times versus 50, or even in multiple locations 50 times different things is different set of data. 100% agreed. All right. That's in the spirit of moving our program along. Chris, I'm going to thank you for your time. And even though making a very big deal out of probably a top 10 word used in the English language, and, right? So really small, big use of a small word today. So thanks for that. Thanks for kind of laying the groundwork for our all 4 of our speakers today, and giving us a kind of an element that we can weave in through all the talks. Really appreciate it.
My pleasure. Thanks, Wes. Thanks for having me.
Okay, so with that, we are going to shift topics, and we're going to go from some of the high-level thinking that Chris and Chris has given us, and Jim has given us, to a more detailed, granular level, specifically for gene therapy. Rachel Legman is the director of gene therapy technologies here at Repligen. And for the next 30 to 35 minutes, Rachel's going to walk us through essentially how multiple technologies at Repligen can be applied to multiple workflows. And, we don't get to that aspirational and like 4 or 5 ants of Chris, but it's really the intent in Rachel's presentation to show you that it's more than one, right? Maybe it's two, sometimes it's three, sometimes in the best case scenario, it's four, right? We don't get all 6 or 7 that we actually need right now, but it's a step in the right direction. So with that, I give you Rachel Legman.
Hello, my name is Rachel Legman, and I'm the Director of Technology Gene Therapy at Repligen. During my talk, I'll present to you the next generation viral vector manufacturing 2.0 at scale with Repligen differentiated solution. In spite of the economic challenges that we faced during COVID-19, the year of 2020 is known as the record-breaking year in various gene therapy modalities. Started from AAV, lentivirus, and end up with plasmid, DNA, and mRNA. So what are the driving force behind the golden time for cell and gene therapy? There is an increased support from regulatory bodies for product approval. There are 6 approved commercialized gene and cell therapy drugs during the last year. There are strong pipeline. More than 1,200 drugs for various indications are in different phases of clinical trial, so we do have a strong future. And therapeutic developers raised $19.9 billion in 2020 in compared to $9.8 billion during 2019. So there is a strong investment going toward this direction. So how can we make the global gene therapy a success story? And in order to make it, we have to overcome 4 main manufacturing challenges. First, meeting the demand. So we need to intensify the vector production through the entire process. Second, we have to meet the product safety and quality. This is critical. We cannot afford not to have a safe product. For that, we have to integrate certain solutions. Speed is the third challenge. So we have to reduce the time to market and meet the demand as fast as possible. The fourth challenge is the cost. So basically, we want to reach the inspirational goal for Chris Lab that we want to have a large amount of viral vector. We want to have a safety vector. We want to have a rapid process, and we want it to be affordable to everyone. But we have to be realistic. So in order to do that, because of the complexity of the product and the complexity of the process in gene therapy, different modalities, we will demonstrate to you how we can bring the manufacturing one that is lacking the productivity and scalability due to the nature of the process and the low downstream yield recovery by integrating some of Repligen technology like the perfusion system. So we can enhance the productivity on the upstream side. And we can increase the yield during the downstream side by using different strategies that I will point up later. We can target increasing, and we already demonstrated we can increase the productivity and the downstream yield. But we cannot achieve it for all modalities. We have to do that in a stepwise manner in order to get to the ultimate goal of the overcoming the 4 major challenges I discussed before. In the next few slides, I'm going to talk mainly about the production of lentivirus. Lentivirus is a major challenge in the industry. because it is very sensitive, it is an enveloped virus, and the recovery yield is really low. So there are two elements that I'm going to discuss: the ex vivo, that is not required a large volume, and the trend of going with a lentivirus in vivo. And that's what I'm going to show you, demonstrate to you. So in this slide, we are demonstrating the lentivirus manufacturing 2.0 for 2,000 liter. So in the upstream side, by using the TFDF system, we are increasing, intensify the cell density. So we are managing so far to overcome this challenge of C-trend simplification by enhancing the cell density. During the production, we also need to increase the cell density to a higher cell density in order to produce more viral vector during the transfection process that require for this process. So we managed to do the simplification and the intensification of the cells during the production. We also are going to show you that by doing multiple harvest or continuous harvest, since the lentivirus is a budding virus from the cells, and by using multiple harvests, you can increase the amount of titer per batch. So only on the upstream side, we are showing you that by combining enhancement of all these steps, we can increase the productivity. But at the point that we can do that stepwise, so people can try to simplify the process, they can try to enhance the viral vector, and eventually reach to the demand, the need for this viral vector. During the clarification, we are continuously using the TFDF, which is transfer the molecules of the lentivirus into the cross-flow TFF in order to reduce the volume of the harvest. In this case, we have to do optimization on the purification step by the chromatographic OPUS pre-pack colon and move into the following step of the purification. And the last step is meeting the product quality, either using hollow fiber with different cutoff in order to optimize this process and meet the safety requirement and the demand that is needed. So overall, I'm showing you here a 2,000 liter that by stepwise optimization, both on the upstream and the downstream, you can reach the goal of overcoming these challenges with the lentivirus production. TFDF system was developed as an integrated solution consistent of filter. So here the cells are using the benefit of the tangential flow going through a wide lumen surrounded by defiltration to transfer with minimum loss of the product through the defiltration. We also have to make sure the process is closed and aseptic sterile by using the ProConnect TFDF 4+ and controlling and monitor the process with a controller system that we have here. So the system can evaluate it for lentivirus production in a small scale that later can be scalable to 2,000 liter. In this slide, I just want to show you how you can try to overcome all the need for the demand for this process by different elements. Either you are going by scale and you're going from 20 liter to 2,000 liter, but we know that we have to compromise on the cost in this case, or you can use step optimization on the upstream production by using the perfusion system to intensify the cells and to doing a multiple harvest for lentivirus. So you can increase the productivity on the upstream side. You can do also increase of the recovery yield by using the optimization on the downstream side. So balancing between all these parameters can maybe bring you eventually to the optimized goal of having lentivirus, which is affordable and meet the demands and high quality. In this case, I can show you that if you are doing the gene therapy 2.0, eventually overcoming all the challenges, you can increase your amount of demand by reducing the cost, by reducing the volume of the bioreactor. So this is a few elements that you can play along the process. One of the case studies just to show you how the perfusion system ATF increasing the cell density from 1.3 million to 12.6 million by 10. We have to remember that each clone of HEC293 is different, so the challenge is still there. There are some cells that can reach in the batch 5 to 6 million, and then we have to find the optimum condition that we can still transfect large scale of high cell density. So this is still work in process, but we can show that in some cases we managed to overcome that. But we have to be patient with many other processes. The other step that people are starting to do, and it's very successful across the board with lentivirus, is doing multiple harvests, because the lentivirus is a budding virus that go outside the cells and the cells remain active and continue to produce the virus. And in this case, by using TFDF, that is very gentle on the cells and on the lentivirus product, we are replacing the defiltration during the harvest with TFDF in clarification mode, and we are harvesting the first time. Then, since the cells are still intact and viable, we continue the process for 1 more day or 1.5 more day and harvest the second time. So just by doing this harvest strategy, by using the perfusion system during the harvest for lentivirus, we are increasing the yield of the viral vector by 200%. So this is a significant step to lead us to where we want to go eventually with the gene therapy product. In this scenario, I'm going to show you how we can, by process intensification, reduce the cost of the process and at the same time increase the number of doses per batch. So in here we are showing you 4 different process scenarios. So the left one is a control scenario that you are using the current process when the cells are not growing to a high cell density, so you need large volume of citrate going to 2,000 liters. The cells grow to 2 million cells and you're using defiltration for harvest. This is our control for the calculation. The second scenario is just changing the defiltration with TFDF that is gentle both on the cells and the virus. The third scenario is doing multiple harvest using the TFDF, harvest 1 and harvest 2. And the fourth scenario, this is the ultimate one that we want to reach eventually after all this step optimization, is decreasing significantly the citrate decrease the volume of the manufacturing bioreactor, intensify the cells to 20 million, doing multiple continuous harvest using the TFDF system. So you can see that there is a significant reduction of cost during the intensification process for lentivirus in this case. I shift gears and talk about AAV. So this just shows you the complexity of the molecules that we are dealing with. So that's why the process for gene therapy is very complex, because it depends what serotype we are using, what transgene we are using, and so on. So in this case, I'm taking a scenario of high demand for AAV, about 5E14 viral vector per dose, and you can see that per year. So you can see that if you need to, for example, address 50,000 patients, you need 42,000 liter bioreactor. So how can we do that? So in this workflow, we understand the complexity of the process. We understand the complexity of, in this case, the AAV. The AAV is more robust than the lentivirus. Therefore, we can use another perfusion system. So on a small scale version, you can evaluate side by side the perfusion system ATF and the perfusion system TFDF for overcoming the CTRAN challenge, overcoming the low cell density at time of transfection, so we can enhance the cell density. So in this case, it's exactly the same thing as HEC293 growing lentivirus before you are starting the transfection. Another point that is critical to improve the process in this case is doing manipulation and different strategy on the media in order to gain best transfection. As of today, without transfection, without perfusion system, using transient transfection process expression system, you are using the same media going to the transfection. That may be the root cause of why the cells are not producing enough viral vector. So in this case, we are showing you how you can improve each step either separately or together in order to enhance your viral vector. At the time of lysis, as long as the product, in this case AAV, is associated with the cells, we must do a lysis. So it will be beneficial to use the either perfusion system in order to reduce the volume before performing the lysis. After the lysis, we have a very high turbidity, high viscosity material that, using TFDF as first clarification step, gain you high recovery of your virus in a fast manner because of using of the clarification. And in this case, you also make another improvement that brings you forward to your overall goal, is reducing significantly the footprint of your manufacturing suite because of the efficiency of the TFDF as a clarification system with using a very low surface area for large volume of bioreactor. After that, you are going into the concentration of the volume further by using in large scale the ARTSYNS TFF system with a flat sheet cassette for the AAV product. And this system has a very low hold up volume. So this is another step that you can improve your yield moving forward on the downstream side on the purification. The OPUS system improve the optimized condition that bring you high yield using the affinity colon, follow up with anion exchange colon to separate between an empty and full. The ARTSYN chromatographic system with a dual UV system and a very low hold up volume and a very gentle pump can increase the yield of your optimized condition bring you to a better place. And meeting the product quality in the last step is also very critical. So in here, I just want to focus on the benefit, one step that we demonstrate that improving, simplify the C-train using perfusion mode. So when you are using perfusion mode, you are increasing the cell density. For C-train, you can go into a very high cell density and therefore dilute and moving into the production scale with a very small volume. So this can reduce the cost. So at the moment you understand that each step of the process can get you to overcome the 4 major challenges that we talked previously. By using the intensification of the cells at time of transfection, we increase, of course, the amount of viral vector produced by the cells. And also sometimes we can improve by perfusion system the specific productivity. So we can make the cells produce more viral vector per cell. But that's something that we have to optimize for various cell lines and various products and various transgen for different indications. So we are still not there, but we are getting there. In the downstream side, we have also a major challenge, but what I want to make sure that everybody understands is the recovery yield is low, so we have to further optimize each step of the downstream process with a good vision of what we are doing during the process. So we must have a monitoring and better control on the optimization development process in a small scale. Once we identify the best condition, we are offering you the large scale, both skid and consumable to reach your target. In this slide, it is very critical to understand that each step on the downstream process can be improved because each one of them show losses of the recovery yield of the product. So by optimization of the TFF system and optimization of the affinity chromatographic, both steps, we can enhance the recovery yield of the product in place. The first selection of TFF format is critical. So based on the product you are dealing with, whether it's lentivirus, AAV, oncolytic virus, and so on, you are using either hollow fiber or flat sheet cassette. The second screening method is using molecular weight cutoff. It's not only improving it's not only concentration and diafiltration, we also have to get added value of impurity removal. So that's why you have to evaluate it, and this will bring you a step forward into a better recovery of your product by reducing process steps. And the final is flux optimization in order to help you to scale up the process with the right condition and with the right system. As I mentioned before, OPUS prepacked columns help you identify the best condition by, as you can see, there are so many factors that can impact your process optimization. And since people do not have a high throughput tools, they are compromised on what are the conditions to bring them as fast as possible to the market. So we know that time to market is important, but recovery yield meet the demand is also critical. So we have to make a decision what is better for us at this point. So by using the high throughput RoboColon, we can identify the best condition to give us the best test product in terms of if we want to separate between empty and full or other product related impurities, and then scale it up to the large scale and make sure that we meet the demands. But at this point, we may not meet the timeline. So at this point, we have to choose our battle. So this will bring you to a better process, but you have to be patient and wait a little bit longer. So this is a strategy decision for each company to make. As I mentioned before, mitigating the risk for contamination is critical. So we must work in automated closed aseptic system, and we have all this product to support you getting and mitigate your risk of contamination. The last point, and not less important, is the use of VPE analytical tools in order to be able online to measure the quality of your product, to make sure the product is high quality and the lot-to-lot consistency is good. So by eliminating the time and the steps that needed in order to evaluate your product is very critical. So this is one element, additional element that help you to get into your process at a time manner and in a good shape. Thank you very much for your attention, and I'll open the floor for additional questions. Thank you very much.
Okay, thank you, Rachel. As promised, Rachel has walked us through the 2 different gene therapy workflows and hit on the multiple ands that Chris emphasized, specifically capacity, but you can also have the cost and in there as well, right? Rachel, we have 4 questions. Just got another one. I think we're going to take these in chronological order, and then I might jump around afterwards. The first one is from Finn. Do you have a model that estimates total volumetric output when each tech is applied? Probably each technology applied. That's probably from earlier in your presentation when you were doing the lentiviral presentation with the multiple, like the different ATF and the different TFDF throughout the workflow.
Yeah, that's an excellent point, because we don't model for each scenario, because we have to remember that even if we are using one modality like lentivirus, we have to remember that it is different. Depends what is the transgene inside. Is the lentivirus include target gene? It's create a lot of complexity on the surface of the virus on top of the other complexity. But we do have a certain parameter that we are taking into consideration, into account. All the consumable is used, the per process, all the media used. In this current process, unfortunately, we are still using the transient transfection, so you have to take into consideration all the elements that you have to use in the process, the plasmids, the transfection reagent. So in principle, you can use this basic model, but you have to modify that based on your tailored process for your product. And if you move into another modality, of course, it is different as well. I hope I answered this question. It's a complicated one. Yeah.
I think that's the answer. It's complicated, right? Because, and I think you can see that in Chris's talk too, is that I think sometimes when you modify the workflow, you introduce a new technology, you can actually create complexity elsewhere. And sometimes you introduce a technology you're targeted in one space and you create simplifications elsewhere. It depends on the process and the technology, all of it, but it requires analysis.
And if I may add a little bit additional point, that you have the current process, so you have to put that in your model. So if you want to increase your performance of your process by reducing the cost and at the same time enhance your potent product, then you have to consider what technology, what new technology in the market exists to get you to this endpoint that you want to reach to. So in this case, if you use an intensify like perfusion system, both on the upstream and downstream, that's already will add another dimension to the modeling of the process that you are making. But basically, we do have a basic model. You just have to tailor it to where you want to get into and what are you doing currently, what step you're doing currently.
Yep. Yeah. Understood. So we have the next question is from Lance. And the question is, how was plasmid degradation controlled at the 2,000 liter scale? I'm going to guess by the timing of it was also during the lentiviral part of the presentations rather than the AAV.
I would say both, because as we mentioned before, the majority of the market for gene therapy, whether it's AAV, lentivirus, any other modalities like even oncolytic virus, are using multiple plasmid in order to do the transient transfection. So we are not there in terms of the biology having a stable cell line. So I would say that scalability in terms of the plasmid is a very critical point. And this question that Lance raised is very important one, because when you are doing the transient transfection in a very small scale in the lab, or even you're doing it in 50 liter, it's very easy. The dynamic of adding the transfection reagent with multiple plasmid, and then you have to deliver this complex very rapidly into the bioreactor, so it will transfect each cell that in order to get a good transfection efficiency. It's not that complicated when you're doing that in a small scale. So he raised the point that when you are doing scale up, this is a major challenge that we are facing in the gene therapy. So you have to make sure that you are working with the right technology, the right differentiated technology that are not breaking your complex, because at this point you are targeting to have all your plasmid within the transfection reagent. So you have a complex of DNA and transfection reagent. So you have to make sure that you are using the right pumps that are not going to kill the complex and let the DNA get out of it and get degraded. So you have to do it rapidly. Even though you are dealing with 80 liter going into the 2,000 liter, and you have to do that very gentle using the right pump. So when you are choosing a technology for your process, it's not enough also to choose the consumable. You also have to make a very smart choice of the technology that is taking into consideration this complexity. So we already managed to scale up the process at least to 500 liter without getting any loss of the transfection efficiency. So it was the same efficiency as we did in a flask or 50 liter bioreactor or 200 liter bioreactor. So you have to break down the elements that you are talking. So you're talking about plasmid. You have to talk about also complex size. So there are a lot of elements in this equation that you have to take into consideration to make the outcome successful and scale up linear your process in terms of the volumetric outcome of your titer.
Okay. Yep. Abs 100%. Rachel, we're gonna stay with Lance for his second question, which is: has the scale of production been performed at scale, or is it theoretical output? Again, I think we're talking about lentivirus. It's great with the other questions. They say LV or AAV. I think he's talking about lentivirus and TFDF. Maybe we'll use that as an example.
Yeah, so for the case, if I understand correctly, Lance, he's talking about the slide that I showed, the economic model. So this is a theoretical calculation based on what we did in a small scale. So definitely we have to do the 2000 liter calculation once we run the process itself. But for this slide, it was theoretical calculation based on the small scale.
Okay.
And since we have a scalability, I'm not concerned about that. But to the question is yes.
Okay. The next one we're going to turn to is from Derek. Is there a reduced yield in the second harvest for lentivirus? Did you, and then a follow-up right next to that, did you ever try multiple harvests for AAV?
Okay, very interesting question and good one. So thank you, Derek, for that. We didn't see any reduction, but I didn't run multiple processes. We did it for, I think, through customer at least, it was pretty successful at least 5 times. So the second harvest is usually you're getting the same potent lentivirus yield as you're getting at the first harvest. Of course, you have to take into consideration the standard deviation of the cell-based assay, the transduction unit assay. But we even try a little bit farther. So there are stable cell lines that you can go even up to 6 days without compromise on the yield. So the cells, as long as they are metal viable, and in the TFDF system is very gentle on the cells, so the cells maintain viable and you nourish them along the process till they cannot take any longer the fact that the lentivirus is budding out. So we manage after 6 days for stable cell line to get each day the same productivity. For transient transfection, on the third harvest, you start getting heat, and also you have to take into consideration the additional impurities that you are enhancing as the process is moving forward. So on the third harvest, you get a lot of non-product related impurities that complicate your downstream, and overall the process you are losing yield. In terms of the second part of your question about the AAV, it all depends what AAV. Most of the AAV currently in the market are associated with the cell. So you must lyse the cells. You cannot do a continuous harvest because the virus is not budding out. You can do that on lentivirus, you can do that on exosome, anything that is continuously coming out from the cells, you can do continuous harvest. But you have to understand the AAV dynamic in your process. If the AAV is excreted from your cell to the spent media, and that's what you're collecting, yes, you can use the TFDF for that as well to do continuous harvest. But unfortunately, the limitation of most customer is that in many serotype is the AAV is still associated with the cell, and we must do harvest. So we cannot do continuous harvest. You have to do a recirculation, no continuous during the production. You no need to do that.
Yep. Bottom line, it's different, right? Lentivirus and AAV are different, and even depends on the AAV. Okay.
Yeah.
Different. Yeah. We're gonna hold on to Hexion. I see Hexion's question on upstream. We're gonna hold on to that for 1 second. I'll tell you why in a minute. And Rachel, we're gonna switch gears and we're gonna go to downstream. Thomas asked the question: have you tried multiple cycles for the chromatography column step, or is the column single use?
Well, it's recommended to be single use, but you can reuse it, but you have to validate each time that the performance is the same. In the gene therapy specifically, we are highly recommend to use the colon one time. And I assume they are talking about the chrome, right? So the OPUS pre-pack colon is accommodating multiple resin from the market. So basically, you can follow the recommendation of the vendor, how many time and what condition to use on your colon. But this is something that you have to put under risk mitigation analysis. And maybe during the development, I know people are doing that, but at a certain point when you do the conformation run, I do recommend not to add additional parameter or variability into the process. So in principle, it's single use, but people doing it multiple times. I hope that's answer your question, even though you don't like that.
I think so. I think they understand. Okay, Rachel, last question. Apologize, Finn. We'll have somebody follow up with you. But from Axion, and Rachel, we're going to use this as a bridge over to Charles' talk, which has a heavy influence, focus on upstream and ATF. We're going to use this as a bridge. Have you tried to achieve higher cell density for USP more than 5x? And if so, what's the highest you can achieve? Have you ever observed an increase in residuals?
Yeah, so that's why I was talking about stepwise improvement. So that in principle, you can enhance. So it all depends what is the baseline. With one customer that we show you the case study, we started their HEC293 in a batch mode, grow at 1 million. So this is in principle very low cell density at time of transfection. But when we enhance it more than 5x, in this case it was 10x, we reached 12 million. We managed to enhance intensify the viral vector based on the number of cells, and more of that, it's also the specific productivity because the environment in the cell was better. But you have to be very careful because in this case study, they were not using a toxic transgene because the transgene is also make a lot of impact here. And if you have as many customers currently in the market, because this dynamic market is moving fast, very every day, every moment as we speak. So some customer in a batch mode can reach 5 million. So I don't have any information that we can increase the cell density above 12 million in order to reach a better outcome in terms of the title that you are getting. So you have to remember the volume, as long as you're doing transient transfection, the volume of the bioreactor maintained the same. So you have the toxicity of the plasmid, you have the toxicity of the transfection reagent to consider, and you have to take into consideration that this HEC293 at high cell density tend to clump. So you have to add a surfactant element to the media to prevent this clumping. This surfactant element prevent from transient transfection. So I cannot answer fully to this question that in some cases it is yes, and in some cases it is not. So it all tailored to the process itself. That's the complexity and the challenges that we are facing with this market, this modality of AAV, lentivirus, oncolytic virus, exosome, anything that you produce by the cell line.
Agreed. And I think that's the challenge. I think the processes around gene therapy are so new, it's hard to have those guidelines. And as mentioned, so our next presentation is focused on ATF as a technology and its application specific case studies with Charles Hill. And I think there's enough of those processes have been brought to maturity where there's no promise that a viable cell density for a specific process, specifically a CHO, is going to reach a certain one. But there are sufficient examples, both in the literature as well as just in general experience, that there are estimates, right? There is a general guideline of what you can expect when you apply a cell retention device to a bioreactor using current supplements, current feed strategies. So Rachel, I want to really thank you for joining us and walking us through the gene therapy workflows, both Lenti and AAV, and hitting on the multiple ands of capacity, flexibility, and cost reduction to a T, like multiple ands across multiple workflows. Thank you.
It was my pleasure, Wes. Thank you.
Okay, so the program is built again towards capacity and also moving from higher level thoughts, broad speaking concepts with Chris, broad strategies with Jim, multiple workflow level perspectives with Rachel. And now we're going to move down a level in detail with Charles Hill, who's an FAS here at Repligen. He works every day with customers, hands-on, boots on the ground, to develop intensification processes, bring them, to the awareness level of the ATF technology, all the way to a validated process of the bioreactor. And because the theme here is capacity, Charles and I have talked, and we've skipped over the idea that of what ATF is, the technology. We have skipped over even the idea of what's viable cell density and feed strategies and all that, and taken it just to the benefit of capacity. So let's hand it off to Charles Hill, who will walk us through a couple different case studies around ATF specifically targeted to increasing capacity on the upstream side.
Well, thank you, Wes, for the introduction. So my name is Charles Hill. I'm an upstream field application scientist for Repligen. And today I'm going to talk about some case studies looking at expanding upstream capacity through cell retention-based intensification. And hopefully you'll see some points I'll touch on in terms of benefits it can bring in terms of reducing the time it takes to produce more product, as well as saving yourselves costs, as well as providing flexibility for your facilities, as well as simplicity. And I think Professor Love did a great job talking about how those ands can be included when it comes to next-gen manufacturing. And I hope some of these case studies will kind of highlight that for you today. Start off looking into increasing capacity with a cell retention device. So some of the big benefits you'll see is being able to increase your overall volumetric output. And that can be kind of done twofold. So first, we'll start off with being able to achieve more cells in a faster time period. That really allows you to increase the overall throughput for your reactors. And then the other benefit is looking into bioreactor utilization. So when it comes to not only getting more product faster out of your end stage, but also kind of simplifying and being able to intensify from vial to end stage, whether that's N-1 intensification or whether it's intensification in your end stage. So why alternating tensile flow, or ATF, works better for these types of applications. So just going into alternating flow, providing a cleaner filter at higher cell densities. Some of the benefits here you'll see with an ATF technology, there's an exhaust and a pressure stroke that is incorporated into each cycle. And during each stroke, half of the filter is being utilized for filtration, whereas the other half is being utilized for backflush, which pulls apart components that potentially could be foulenced from the membrane wall and allowing it to be cleaned through each stroke. And then through a full cycle of exhaust and pressure stroke, you're getting a full, 100% utilization of the filter for either backflush or filtration during each cycle. And that's really what's allowing you to achieve these higher cell densities that can improve your throughput. So we're going to start off with kind of the big picture where we're trying to get at in terms of what the ATF can provide from a facility level decision. So some of the big benefits you'll see in terms of being able to get more product in a faster time is it really helps you rethink the idea of your facility fit. So when it comes to incorporating an intensified process, instead of looking at a larger bioreactor scale, let's take 5,000 liters as an example, you can get away with a smaller footprint, let's say 500 liter, when incorporating the ATF technology. So it allows you to reduce that overall footprint in your facility, giving you flexibility while still maintaining the same amount of throughput. And then also you're able to achieve more through each run and allowing you to actually get more runs per year per bioreactor, which again is helping that increased capacity. And then finally, just looking at a cost perspective, again, being able to reduce that bioreactor size, you're seeing a significant decrease in CapEx expenses. And kind of highlighted below on the slide, you'll see is, as an example of what the ATF can do from a facility level decision, is you're looking at, anywhere from a fourth of CapEx to a third of OpEx, as well as reducing your footprint, it could be up to 10x, as well as reducing the time it takes to produce that same product. So a lot of people, when they see, a cell retention device application, they generally have a connotation that it has to do with, more traditional perfusion or steady state perfusion. But what we really want to highlight here is how you can intensify, even a current fed batch user's process. And so you may be saying, how can we do that without implementing a continuous type process? But I hope some of these future, case studies that I'll cover today can kind of show you where you can use a cell retention device to not only be incorporated in a typical perfusion process, but also intensify a current fed batch process to really achieve those goals of producing more product in a faster time period at a lower cost. So to start off, there's two main applications I'll highlight here for using the ATF technology to increase your throughput and yield. The first one is N-1 intensification. So the big goal here is we're really trying to decrease the production time in end stage while still maintaining the same productivity so that you can achieve more batches and throughput in a given year versus your current process. And so really it's just highlighting on the fact that you're able to achieve, growing more cells faster and allowing you to get overall more throughput in your facility, whether that's increasing the amount of campaigns that go through, the amount of molecules you have in your pipeline for clinical manufacturing. That's really where this application is coming into play. The second one is looking at high productivity harvest. And so this one we're really focusing on, maintaining the same batch duration that you would typically see in a fed batch process, but typically around the peak VCD of a process, we'll start a continuous harvest or a perfusion type application for the rest of the run to continuously, clarify material, as well as give you the benefits of getting more product in that same time duration. So again, just looking at more product faster, that's kind of some of the high level benefits you would see from adopting a high productivity harvest type application. The first case study I'll go through is a paper that was actually recently published in 2020 that is looking at incorporating an N-1 perfusion seed to intensify a current fed batch process. And what you'll see from this paper is that they went from a process B to a process C that in the process C incorporated a perfusion N-1 to grow to higher cell densities so that they could seed their production or end stage reactor at a higher cell density at around 10 to 20 million cells per mil. And after optimization work was completed, they were able to see the same amount of product they would get in their current process B in half the amount of time, or they were also able to get a 2x increase in overall titer when in comparison to their process B. And with those levels of titers, it was also, as high as industry's highest reported values, so around 10 grams per liter, while harvesting in the same 14-day process like their current fed batch process. So it really shows you benefit in terms of, again, getting more product in the same amount of time, really helping to speed up, the capacity for our facility by improving that throughput. The next case study looks into, again, kind of an N-1 perfusion application, and this paper was published back in 2014, and it's looking at, removing or shifting that growth phase in the production stage into the N-1. So what they did is they used a N-1 perfusion seed with the ATF technology and grew their cell density up to 40 million cells per mil in their N-1 stage, and they were able to seed their production reactor at 10 million cells per mil. Now the benefit at, shifting that growth phase from the production stage into the N-1, they're able to improve their volumetric throughput for each reactor. And as you can see in the chart below, they were able to get, greater than 30% capacity when it came to using the high cell density fed batch versus their current platform. So again, just showing the benefits of, being able to get more products faster is really highlighted by this case study. The final major case study that I'll get into for N-1 intensification is, again, another paper that was published in 2020, looking at, different types of upstream and downstream process improvements, whereas the final process they kind of landed on was using a perfusion N-1 tank with an ATF, and they were able to, from their process A to process C, able to get to, over 100 million cells per mil in their N-1 stage, which allowed them to, increase that starting cell density in end stage up to, between 10 and 20 million cells per mil. And the benefits they saw here, as you kind of see the progression from their process A, B, and C, is overall they're just getting more product in a faster amount of time and really kind of, helping reduce that cost by getting the more product per batch. And as you can tell from process A to B, they were able to get a 4-fold increase, and then from process A to C, there was an 8-fold increase in titer. And some of the main highlights here, and from a cost basis, if you look on the bottom left of the slide, you'll see an actual cost analysis that was performed for each process. And you can see up to a 10-fold reduction in cost when it came from process A to process C, really highlighting the benefits of promoting kind of a next generation manufacturing platform to reduce the cost for producing these therapeutics. So this next application, again, is going back over the high productivity harvest. So this was a customer success story using an HPH method and comparing it to their current fed batch process. So what you'll see in the BCD graphs is, once it reached the peak cell density around 4 days into the process, they started a perfusion type process from day 4 to day 8, and they were able to increase that cell density 4x from what they currently get in their fed batch process while still maintaining high viability near 100%. And the benefits in terms of the overall product that was produced, when comparing it to the same duration as their fed batch, they were able to get 2x increase in total protein produced for that process. And not only is that an increase in throughput for that reactor, but it's also simplifying the process because now they're removing a clarification step that would typically be done with their fed batch process, since that cell retention device of the ATF is being used to clarify it down to a 0.2 micron cutoff. So again, it's kind of a twofold benefit here in terms of getting more throughput, but it's also simplifying their process. So just kind of a high-level overview of what each of these applications provide as a benefit for different steps in the process. For the N-1 intensification, we're really looking at boosting throughput in a facility, and the big areas where you'll see this impact the most is going to be your clinical manufacturing and commercial manufacturing spaces. So you can think from a clinical manufacturing space, trying to get as much product created to get to clinical trials as fast as possible is important, and being able to have this increased throughput through an N-1 perfusion type application is really allowing you to get more molecules through that clinical manufacturing facility faster. And that's a huge thing with companies, is trying to, get that pipeline available to clinical trials as fast as they can. And then from the commercial manufacturing space, once a product is approved, being able to get, the most product in the fastest amount of time with the lowest cost is really where you see a benefit here by incorporating that N-1 perfusion. So for the second one, looking at the HVH in terms of the benefits it brings, the big benefit here is, again, getting more product per run. And so what you really see is an improvement in productivity. And again, that helps out with, clinical manufacturing as well as commercial manufacturing when it comes to, being able to conduct less runs because you're able to achieve more product, or you're able to just achieve more product from the same amount of runs and being able to get a larger patient pool access to your therapeutic. So that's kind of the high-level benefits that each of these applications can bring, specifically kind of towards the clinical and commercial manufacturing space. So this slide kind of provides an example of some large ATF installation sites globally, just kind of some benefits that these customers have seen. And so to start off with, we have Transcenta and Sanofi, who both have integrated continuous processes that are set up in their facilities. And you can see with the Transcenta example that they were able to, get greater than a metric ton of drug substance through a facility with a design process and implementation that took less than 18 months. So that's really kind of harping on the speed that is required for these types of processes, and showing you that this isn't making a process more complicated. It's actually just as simple as what you would currently be operating with. And then the two other examples on the bottom, so WuXi, for instance, was able to intensify, 5 to 6% of their reactors and improve, 50% of their capacity through the use of a cell retention device. So again, just showing that, the flexibility to be able to just intensify a small set of your reactors and still get that huge capacity increase is huge. The last part is just the Samsung Biologics facility in Korea that was adopting an N-1 perfusion at a 3,000 liter scale. And the big highlight here is, they were able to reduce their production time by 30% with the incorporation of an N-1 seed. So again, this is just kind of going off of what's important for next generation manufacturing: speed, cost. These all play into that factor. And then the last example or application I'll get into is kind of where can this technology take you? And for, in terms of some other aspects of next-gen manufacturing that's important, things like simplicity, flexibility that, Professor Love mentioned during his presentation. An example of what, can achieve that is being able to facilitate a smaller and faster manufacturing platform by using a frozen intermediate, such as using an ATF device to grow cells and freeze cells in process intermediates that can really shorten that C train when it comes to a production facility. And so instead of having, 10, 12 steps in an upstream process, you now be able to, reduce that to 3 steps, whether it's just the FOG, the frozen intermediate, connecting it with an N-1 perfusion tank, and then going directly into your end stage. So we're really simplifying the process down to 3 steps, and also providing you flexibility now that you're able to start and stop runs in a shorter time frame, and giving you the ability to get multiple products, multiple campaigns through a same facility in a shorter amount of time. And so that's just kind of where the technology can go, where it can kind of hit some of those targets for next-gen manufacturing to improve your capacity by, again, kind of touching on decreasing, the costs for your processes, giving yourself flexibility, simplicity, as well as just speeding up the time it takes to hit that capacity increase. With all that being said, thank you all for the opportunity to discuss some applications that could benefit each of your work zones, and I'm happy to take any questions. I'll turn it over to Wes to facilitate a Q&A session. Thank you.
Okay, thank you, Charles. So again, we've kind of continued our march towards multiple ends, right? Like Charles, once again, more capacity and cost reduction. I think, especially on that last note, right? And flexibility or simplicity. I think especially the simplicity in that last slide. Um, because we ran out of time on the Q&A of Rachel's part, I took a question. I did push a question well, that we'll answer later. But then during Charles's talk, I said, I'm going to bring that one back because we're so focused on bioreactor intensification during your talk. So Charles, I'm going to give you the question: does bioreactor intensification impact flexibility?
Yeah, no, I think this is a great question, especially because it can impact flexibility in a multitude of ways, whether it's decreasing the footprint of your reactor size, giving your manufacturing facility more flexibility, whether that's being able to fit more technologies in a smaller space, kind of as Professor Love mentioned, getting that volumetric throughput tied in with cubic meters of facility. So I definitely, that helps out in flexibility there. But also whether it's, being able to run a full pipeline of molecules through a clinical manufacturing setting in, a short amount of time, being able to switch from, one process to the next in the same facility is definitely provides flexibility as well. So I think there's a few aspects that definitely provide flexibility when it comes to using a cell retention device for intensifying your bioreactor.
Yeah, I do. So and like if we continue to kind of connect across presentations, right? I also want to go back to Hexion. I know Hexion's question was probably specific to gene therapy, but you probably work with both gene therapy customers as well as kind of traditional MAB customers. But maybe since your case studies, okay, so maybe this would give people an idea of where gene therapy is going to be in 5 years. Could you just kind of speak to a couple different scenarios, because we have so much data, about what are reasonable expectations for the CHO monoclonal recombinant protein side?
Yeah, no, I think it's great. I think, if you look back earlier processes using kind of a cell retention device for either perfusion or a bioreactor type intensification process, it started somewhere, right? So generally you're looking at kind of lower cell densities in the initial adoption of, using a cell retention device, whether it was at 20 to 40 million cells per mil as kind of a starting place for, protein production. But now, we have very advanced users who've come a long way through whether it's developing media, their overall cell line, whether it's their bioreactor parameters. And, we have customers that are able to achieve up to or even greater than 200 million cells per mil. So kind of as Rachel pointed to, it's kind of a stepwise progression. You're not going to go straight from, 10 million cells per mil straight to 200. But it's just an idea of where the technology can take you. And something great about using people like field application scientists and so on is they provide kind of that consultation that you may need to see where things have been, where the process can actually go, and provide feedback on kind of the background we've accumulated when it comes to a cell retention type device. So I definitely, as a starting place, you can get very high cell densities, but again, it comes down to where your current process is at, what kind of steps you can include that give you benefits in terms of flexibility, cost, productivity, things like that, and whether you need to slowly kind of work your way to elevating it to that, kind of pie in the sky dream that actually is achievable, but taking those intermediate steps to reach it is very important.
Yeah. And that's, we didn't touch on it in this seminar series, really. We just focused on like what is achievable in capacity expansion, right? But both, I think if someone's looking at the eightfold, the fourfold increase in capacity, the quarterfold footprint and all that, and they think it's far away, I do encourage them, like just like Charles said, there's a ton of expertise on the team that sees a lot of different processes, so you have access to that. And the ATF technology as a whole has really improved recently. and which has also made it a lot simpler, right? So it's really easier than ever to get there, right?
Yep.
So we did have a kind of a two-part question come in from. I really like the abbreviation of initials these days, so I'm going to go with SD. So the question is from SD. First part of it, like I think they're coupled, so I'm just going to read them straight through. Mm-hmm. From the regulatory point of view, the intermediate product can be stored, or in other words, is there any validated process starting from the high cell density bank bag instead of vial? Probably specific to that last slide where we talk about simplifying the seed train. Yeah.
Yeah, no, it's a great question. To my knowledge, there isn't any current process that's validated that uses that specific high cell density intermediate step, but it's more of an idea of where the actual cell retention device can take you in a process, and just kind of providing a picture of, being able to think of it outside the box and how you could use a cell retention device technology to shorten that, manufacturing time frame and give yourself more flexibility when it comes to manufacturing, whether that's restarting a seed train, lowering the amount of operational time it takes to go from thaw all the way to harvest in the upstream. Those are all important goals, and it just kind of highlights where the technology could take you. But it doesn't necessarily mean it's something that at this moment, or this, moment in time, is heavily adopted or used in a specific GMP validated process. But it's more just picturing where you could take this technology.
Yeah, 100%. All right, that's our 5 minutes. We're up on time there. We've also covered our questions. If you want to reach out to Charles, his email was up there. Have a conversation about intensification on ATF. I'm sure he'd appreciate it. So Charles, thank you for joining us today, and thanks for the awesome presentation.
Thank you, Wes. Thank you.
Okay, so that brings us to the last presentation of the day, and especially around capacity. Again, multiple ands in the next presentation with Danny. But the dots to connect with the next one are, it's not always, if you look at all the needs across the workflow that you see, going back to Chris Love's, right? Not every part of the workflow requires an incredible new technology, filter technology, right? Some of it simply needs development of the supply chain, how we do current things, or leveraging technologies that exist in other markets and bringing them in. So the topic for the next presentation is really single-use flow paths. And Danny has joined us to talk about multiple ands, specifically about, about how Repligen is expanding capacity for its ProConnect single-use flow paths and applying the principle of multiple ands to especially improve the customer experience around accessing these really critical components to create a closed system for gene therapy applications, right? So with that, I give you Daniel Spurgeon, Product Manager for ProConnect.
Thank you, Wes. Thanks, everyone, for having me. I'm very excited today to spend some time to talk to you a little bit about single-use flow paths. My name is Danny Spurgeon. I'm a Senior Product Manager here at Repligen. As Chris mentioned in his talk, even in the design process, it's really important to consider the speed and the cost and the flexibility and the simplicity as crucial considerations when developing product to match to process. Today, I'd like to show you how we've incorporated that into some online tools and some workflows to help develop a single-use flow path around our core technologies. Today's talk is entitled From Design to Delivery, Advancing the Speed, Simplicity, and Manufacturing of Custom Single-Use Flow Paths. When developing a new process, there's a huge amount of variables and considerations one needs to consider. Is there facility modularity? What does your unit operation look like? What scale are you trying to achieve? You may have certain system requirements from existing componentry or new systems that you're planning on purchasing to develop this process on? Are there specific constraints related to that system as it relates to the layout of your suites? What is the core technology that's going to help you achieve your process goals? You're looking at hollow fiber, flat sheet cassette, or novel technologies such as TFDF. Once you've decided on a core technology, there's a multitude of single-use componentry that have to have their own individual considerations to achieve the process-related product you need, whether it be achieving a low holdup volume or having connectivity with sensors to your own system. The list goes on and on, whether it's tubing, tubing length, tubing size, what sort of connectors? Are you dealing with a high pressure environment? Is sterility a requirement for your process? So all of these, it makes it a challenge as to where to start. And in order to get a product that aligns well with process, it's really important that you partner with someone who has the expertise and the workflow to make this as seamless as possible. We at Repligen have a partnered and holistic approach to make this process as easy, simple, and enjoyable as possible. And this slide is really the agenda for this whole talk. We have a core team of experts to help refine those process requirements and start to scope out what process requirements will match a product to align to it. We've worked to roll out some online tools, some technology that will help both inform and walk through that decision-making scheme, and quite quickly develop the engineering drawings and pricing associated with a given product. Repligen has worked tirelessly to make sure that we have supply continuity both into our organization, and we've also invested heavily to expand our capacity to assure supply continuity to your organization. So we're going to blow up each one of these and talk about them in greater detail throughout this presentation. But basically, the ultimate goal here is to take the fuzziness of all of the considerations in the previous slide and talk about how we can make a streamlined development process and the simplest product possible for your process. So it all starts with our consultative approach. We have experts with decades of experience, whether they be application specialists, someone in the field, someone in R&D, someone who's been in the business and systems just initially came up, decades ago. And with all of that expertise comes a series of questions and understanding of what your process requirements are. So ultimately that starts with, well, what's the goal? What's the core technology that's going to achieve that goal? What's your application, and what is the facility fit? That might start to inform some process parameters. Well, if we're trying to achieve these process goals, what are the flow rates and the working volumes and the pressure considerations to achieve those goals? Are there risk mitigation strategies required for GMP, such as integrity testing or sterility? And what scale is this process going to look like? So as we work through this, we're starting to march towards what single-use componentry is going to align to achieve those goals. What aseptic connections are required? What sensing is required for your operation to communicate with your system in a reproducible manner? What sort of vessels do you need for this, whether it be a collection vessel or a processing vessel or a waste container? And what are your holdup volume considerations? Do you need to sacrifice design flexibility for less tubing length? Ultimately, all of this starts to scope out, which we'll look at our process in the next couple of slides, is a engineering document that scopes out both visually and as a bill of material exactly what the product is to align with your process. So this is where we want to talk a little bit about our online configurator tool. This is a tool that we've rolled out as a way to smooth out and rapidly speed up the design process. So you have the great ideas. We've now understood your process requirements. Now we have to figure out the easiest and most seamless way to generate a product. So within this drag and drop environment, we are able to flush out those ideas into a flow path around one of our core technologies and create an accurate technical drawing in an instant. This is counter to traditional processes where you'd have to start with an email or a napkin envelope, or back of the envelope or back of a napkin process, and then start communicating that to engineers that have a blank page. That whole process has now been refined and built into the intelligence of this online tool. So within this digital environment, we are able to build, edit, and save and create rapid drawings. But this is not a start from scratch environment. The conversation that you are having with our experts is also informing the attributes through this process. So what we call our selection wizard starts with those parameters and those goals of what your core technology is, what filters within that core technology, some of those process considerations we were talking about earlier, to ultimately arrive at a jumping off point, fully realized flow path that is within the bounds of the filter technology and the system technology that you've considered. From there, it's just a matter of a drag and drop from a component library here on the right to refine a jumping off point drawing to meet your specific needs. So all of the componentry here, we have over 250 individual components within this library to be able to expand from a simpler drawing here, as you can see in the middle, to something either simpler to align with your process or far more complex to align with your process. As you go through this with our experts, we will start to register the component details here, which ultimately will inform the bill of materials, the preliminary engineering drawings, and the pricing associated with such a flow path. So it's great that we have these digital tools, and it really helps facilitate this process. But this is not just fluid management. We are dealing with multiple differentiated technologies. These core technologies are already inputted into the configurator. Whether it be Spectrum hollow fiber filters, the new and differentiated TF-DF filter, Tangenix flat sheet cassettes, or our line of reservoirs specifically designed for TFF, or even just traditional fluid management tube sets. Spectrum hollow fiber is probably the most commonly paired product with ProConnex. There's a huge variety, large breadth of scale and selection within this high flux hollow fiber TFF membrane. There's 7 different module sizes, 7 different module lengths, multiple chemistries and lumen diameters across 16 molecular weights. So these highly selective molecular weights allow us to select a filter that is very specific to the needs of your process. Pairing those with our series of TFF systems from the KR2i here at the bottom at the lab scale, the KMPI in the middle, the process and the pilot scale, and all the way up to the KTF 1000 and 2000 skid you see at the top of your screen. So within this, I don't want you to think that we are simply taking a filter technology and aligning it with a system. Our goal is not merely to manage fluid through this system, but to build a flow path for the process. And I'm going to go through just a few examples to show that we are really working towards satisfying needs as opposed to satisfying filtration and system requirements. This first one up top you can see is a simpler flow path. This is built around that pilot scale KMPI system, a peristaltic pump. This customer wanted the lowest holdup volume possible and quick disconnect connectors. So you can see that all of the attributes you see and the other ones are paired substantially down in order to align to process. The bottom one here in green, this is a smaller scale process for a KR2i system. However, this customer wanted to achieve multiple flush steps at the beginning of their process that they wanted to connect aseptically, both at the feed of the process and the waste of this process. They also wanted multi-stage collection of their product, and they also wanted the ability to manage fluid and divert fluid manually throughout all of these T's and Y's within this process. On the right, we have another example of a flow path that's far differentiated away from simply managing fluid through a filter. This specific customer wanted multiple waste containers off the permeate. They also wanted to leverage our TFF reservoirs both as a processing and feed reservoir as well as a buffer addition reservoir. Another requirement of theirs is that they required in-process sampling. So we have a needle-free sampling port built within this library. And then they also wanted a separate bottle for final harvest. Another core technology that we released last year that pairs perfectly with ProtoConnects and FlowPath designs is TFDF. So TFDF is an innovative and completely new exclusive Repligen technology. It's a tubular shaped filter, which makes it look a lot more like a hollow fiber. In fact, this housing is the same material of construction as a traditional hollow fiber housing. However, the filter itself has a lumen that's 5 times larger and a wall thickness that's 50 times thicker. This allows this filter, while flowing tangentially for high cell density, it has the high transmission of a depth filter. So you're able to achieve the process step of depth filtration, but wrapped up in a much smaller and user-friendly format. This results in an order of magnitude increase in flux and capacity that turns to reduced costs, substantially reduced complexity, and a 3-time footprint reduction when compared to depth filtration. So the operation is fairly similar to hollow fiber. The feed stream enters the bottom of the lumen and travels upward, recirculating back to a bioreactor. However, the filtration path is of short, highly interconnected paths similar to depth filtration. So as we introduced this technology, we felt due to the process requirements within this customer group, within this application, there are certain specific attributes that a ProConnects flow path is the right format. Of course, when you're starting out at the PD scale, we offer a filter-only version of this. But as you scale up or you require certain reproducibility or you're working directly with one of our KTFDF lab scale products, we offer an off-the-shelf standard version of this version, fully rendered into a fully integrated flow path of ProConnects. However, if your process requirements at the lab scale or as you scale up into the process and pilot scale have specific process requirements that differ from standard, we can also incorporate our digital environment and create a flow path around our KTFDF systems and our TFDF filtration technology to align this product with your process. Either way, whether you're selecting the standard or the fully configured and custom in the digital environment, at the end of the day, you're going to get a closed, single-use, gamma-irradiated flow path. Requires no flushing. It's already incorporated with our systems through integrated sensors and a disposable pump head. Another great way to leverage our process expertise, our digital environment, and our flow path construction is to pair ProConnects flow paths with Tangentix flat sheet cassettes. TangenX flat sheet cassettes are the highest flux in the market, very differentiated, high quality membrane that pairs in two different ways with ProConnex. We can make a fully integrated flow path around the TangenX filter plate insert for easy on-site integration of a CS cassette. Alternately, we can design a flow path around the aseptic connectivity of the CS Gamma device to provide easy aseptic plug-and-play processing on site. So whether it be one of the core technologies from hollow fiber to TFDF to flat sheet cassette, or one of our processing reservoirs, at the end of the day, with our consultative approach and our online environments through the design phase, we ultimately arrive at a formal engineering drawing to represent both what will be manufactured and design lock for a product that is perfectly aligned with your process. So whether it be this highly flexible hollow fiber on the right, top left, or a simple 50-liter processing reservoir on the bottom right, we are now at the point where it's time to make some considerations on how to manufacture such an assembly. So as Dr. Love mentioned in his talk earlier, pandemic-led supply chain instability has had a major impact on our industry's ability to deliver medicines in a timely manner. We at Repligen have worked tirelessly to forge a strong supply chain and risk mitigation across a cross-functional approach and many programs to make sure that we have supply chain continuity. It all begins with the parts. We developed a program we call the Standard Toolbox Program, which allows us to have a bit more of a myopic focus on those components that are within the configurator and commonly used within FlowPaths to allow us to have hold higher stock, supply agreements and quality agreements with those critical components suppliers. So in the event of instability, we have a hedge against such instabilities. Our sourcing teams monitor our sourcing and planning teams monitor our materials for shortages on a daily basis. We evaluate things at a higher level weekly, and we're constantly mining for risks to mitigate through our senior leadership risk mitigation council. We over time have forged extremely close and extremely strong relationships with all of the sub-suppliers to develop a flow path. We're actively engaged with them, and we are constantly developing our relationship and getting a detailed understanding of what is going on within their organizations to better weather the impacts of instability or supply shifts for them. In addition to that, we're always mining for alternatives. Our engineers are always looking to identify and validate suitable and recommended alternatives for any sort of individual components within a flow path. So if any more supply chain instability comes up and the product that was originally designed is not producible because of a stock out or an event on an individual part, that's okay, because we have suitable replacement parts, and under deviation, we can easily produce something that is still meeting the process requirements for the customer. At the end of the day, our goal is supply continuity to ourselves, and we feel with this multi-pronged approach, we can deliver. Now, our ultimate goal is supply continuity to you, the customer. With supply chain security and continuous product flowing into our organization, we still have unprecedented demand as we continue to support the bioindustry's expansion in mRNA vaccine production and gene therapy. Therefore, we've invested very heavily in near-term and long-term expansion plans to produce the multiple ands that Chris was talking about of speed and flexibility and safety and simplicity for our bioprocess customers. To that end, We have rapidly increased our capacity both in our existing facilities as well as an expansion to multi-site capabilities within the next 18 months, ultimately achieving a total of 9x manufacturing capacity compared to last year. With that, I want to thank you for your time, and I will turn it back over to Wes to start the Q&A.
Okay, thanks, Danny. Once again, the big little word. And Danny, I can't stress enough how I love this presentation specifically because I think the "and" kind of sneaks up on you, right? It's about, because how do we build Chris's aspirational goal of technologies that go across all sorts of different modalities, right? With high capacity and do it quickly. So I just think it's a great contrast to ATF, where this is more about say like the customer experience and getting done and leveraging technology specifically.
Sure.
So I'm going to use a segue to go to the first question we have, which is, does the configurator automatically pre-select components that are compatible with each filter? Like
yeah, it's a good question.
Yeah,
it does. It always tries to use intelligence to figure out what the answer would be. So if you're dealing with something of a certain scale, where it's possible, it will configure the components to align to that scale. It uses a very easy red-green color scheme to tell you if it's wrong or right. So if there's as you adjust from those jumping off points into something more related to customer process, and you start making adjustments to that, and there's any sort of place where there is an error or something that doesn't align with that size, it will tell you, and you can easily go in and make sure that it aligns throughout the entire flow path.
Yeah. So could you speak to, so it's not, so even the idea that you have the digital environment and you have the red, green, and I'm assuming like if you have a certain dimension tubing, that it won't allow you to put the wrong size connector on it, right? But I think one thing we didn't do is contrast that to maybe what we did before the configurator, right? Like maybe we did like what we did these things by email or by hand.
Sure.
Could you go back a step and like kind of just highlight like that what a big step that's been?
Yeah, absolutely. Because it was really a shift in how we approach things. So in the more traditional approach, it would be a wide variety of customer inputs. It could be an email. It could be a picture of someone's setup. All the inputs were completely variable, and then handed off to a very talented group of engineers that understand TFF and process really well. But there was sort of a challenge in interpretation and an extremely varied environment, and they're starting on a blank piece of paper to try to create product from whatever the input that was handed to them. So the configurator, because of their experience, we have been able to input some of the business rules that existed in their minds, whether it be flow rate as it relates to tubing ID or durometer of tubing as it relates to a certain system type, and incorporate all of those inputs into the infrastructure of the configurator. So we didn't lose anything from switching from a completely human environment to this digital environment, but what we did gain is a consistent input. So gone is that variety, and the jumping off point is already informed. So when all of this is done and it gets to engineering review, what is done in that digital environment is already translated into a traditional engineering CAD file. So they are working within the same bounds of the configurator and the same inputs that the customer and someone in the field have generated.
Yeah, it's really important. I think it's again, we're kind of playing catch up to some of the other experiences that we have in our everyday lives, like with Amazon or other, but bringing those types of digital experiences into the bioprocess manufacturing space, I think it's phenomenal. It's more like customer experience than technology, but it's equally important.
Sure. Yeah, it does also and has allowed us to scale to work with more customers and create more drawings in rapid sequential for a given process or a given group within a company. So the scale was really important in developing this process.
Okay. We do have a two-part question from DA, which is, I'm just going to read them maybe together. You can handle them together as you wish. Hi,
Sure.
do you, yeah, hi DA, thanks for the greeting. Do you also supply single-use tube manifolds branching off to different points, structural support for proper tube management, and then also what would be the maximum operating pressure for these single-use tube manifolds? Maybe not just the manifold, but the flow paths in general that ProConnex creates.
Sure. Yeah. So we absolutely can make any sort of tubing or fluid management flow path for your process. As far as structural supports, a new part of Repligen Artisan does offer some supportive arms that they developed, a sort of a exoskeleton for tubing that's available. Other ways we can support it is just part of our system setup, and our system skid setup. It kind of organically has that with the bracketing that comes on the skid. Other ways we can have more structure is to use thicker durometer or braided tubing within a flow path, because ultimately the problem is a kink or something that will restrict flow. As far as maximum operating pressure, that's a little bit of a loaded question. So all tubing has certain burst pressures, right? So you could theoretically total up all of the different burst pressures and come up with whatever the rate limiting one is. However, every single connection creates a different environment. Let me give you an example here. Silicone tubing is very robust, but it's very elastic as well. So you can imagine a leak occurring because it expanded over a hose barb. So we don't really offer a maximum. Generally, everything can exist within that 0 to 20 PSI realm. We do offer integrity testing to assure that there are no gross leaks, but ultimately it is operation driven, that if there is something that requires a higher PSI, well, we simply build a flow path that aligns with that better. Braided tubing, we can use Oetikers instead of cable ties to try to create the most robust assembly for an operation, but we can't necessarily produce a generic maximum because of the variability in a custom design.
Okay, understood. So we also have a question about components. Are there automation components related to the configurator, or maybe built inside the configurator, available in the drop-down on the right, as well, like transmitters, force sensors, and control components? I mean, I know we have sensors in that drop-down component, but I don't, yeah.
Yeah, absolutely. Most of the standard toolbox items that are in the configurator are related to communication with our own Repligen systems, right? So pressure sensors, flow sensors, anything related to single use that is aligned with our systems and communicates with our systems already come pre-populated within there. So yes, we do.
Okay. All right. I think that brings us to time, trying to keep our 2021 technical seminar series on schedule. So we're going to wrap up our Q&A. Danny, really, really grateful for you coming and adding the component of leveraging other technologies from other markets, like online configurator, to the customer experience, not just like new fundamental technologies, crucial to achieving the multiple ands.
All right.
So thank you for joining.
Yeah, well, thank you for allowing me to be here.
All right. So as a wrap up, thank you again for attending Repligen's 2021 technical seminar. We started with the aspirational tone of Chris Love that set the stage for the need for capacity and flexibility and cost reduction and safety. And I hope that you recognize through the following presentations that each of the technologies, applications, or even here, the user experiences that are being generated contribute to multiple ends, right? I think the entire industry is working towards that aspirational goal of all of them. But I think if we achieve even 2 or 3, then that's a fantastic win. Jim highlighted the extent that Repligen is leaning in and investing into the capacity challenge with increases in capacity that range anywhere from 3-fold all the way out to 9-fold, for example, with Danny's ProConnex. And then we moved on to Rachel, who walked us through multiple workflows around gene therapy and the ands that are achieved across multiple technologies. Charles walked us through the ands of several different use cases and the order of magnitude increase in capacity at the upstream side. And then Danny on the technology side for single-use flow paths. Once again, thank you for joining our seminar. We look forward to seeing you at another Repligen event in the near future. Take care.